This is a video which looks at the aspects of protein structure which you need to be aware of for your A-level chemistry exam. So here are the learning objectives for this video. We'll have a look at the importance of the sequence of amino acids in determining the structure and hence property of proteins. We'll make certain that you are clear about the difference between primary, secondary and tertiary structures of proteins, and then explain how the various types of intermolecular bonding that you're aware of play a role in determining these various levels of structural features of proteins. So what then is a protein? Well, we're aware that proteins are naturally occurring polymers, and you'll often see diagrams in books looking something like this, and it's clear that what we've got here is an extremely large molecule, but what's the meaning of these various coils and folded sections here, and how does that relate to the chemical structures that make up the protein molecules? Well, we'll have a look at those features in a second. So, a little bit of revision about the molecules that make up proteins, and proteins are formed by amino acid molecules joining together. So here are the 20 naturally occurring amino acid molecules. So we're already familiar with the idea that they contain a central carbon and then a carboxylic acid group and an amine group and a whole variety of different side chains attached to that central carbon. Notice, by the way, that these amino acids are shown in the Zwitter-ion form, which is the form in which you'll usually encounter them in chemical questions. So we can focus in on several different amino acids here. So here are the names of some amino acids and their structures. You can see the side groups there. And we've put together the carboxylic acid group and the amine group of adjacent molecules. Now, under the right conditions, the amine group and the carboxylic acid group, albeit in their ionized forms, can actually react together to produce a peptide link, often called a peptide group, which enables the different amino acids to become joined together. So we have this backbone, essentially, of carbons and nitrogens running through the molecule, and then the side chains are off at right angles to that backbone. Of course, in reality, the chain would be folded and twisted into all kinds of arrangements. Just one comment, by the way, this diagram shows something that is described as a peptide bond. You should be aware that the usual terminology is peptide group, and peptide group includes all four of those atoms: carbon, oxygen, nitrogen and hydrogen. So what we have here is a polymer produced by several monomers joined by peptide groups, and therefore it can be described as a polypeptide polymer. In the previous slide we saw several amino acids joined together by peptide groups, so that was looking at just a small number of amino acids. Here are 4 of them shown in this diagram here. We've omitted the details of the atoms that make them up, because what we're showing here is the way in which quite a large number of amino acids can be joined together in a very long sequence to produce, well, this would be a relatively small protein or polypeptide chain. And it's that sequence of amino acids which is known as the primary structure. So this long line of amino acids forms a primary structure. The sequence of amino acids, therefore, is identical to the idea of primary structure. And it's important to realise that that primary structure is held together by peptide bonds, covalent peptide bonds between the amino acids. So primary structure is the sequence of amino acids within the protein and held together by those covalent peptide bonds. So here's an example of a relatively small protein, one of the smallest of all. In fact, it was the first whose structure was determined by chemical means, insulin. And you can see here that insulin actually consists of 2 polypeptide strands held together by these rather mysterious cross links here. We'll come across those later on. And you can see here the primary structure, the sequence of amino acids that make up these 2 chains. Now, even with simply 40 odd amino acids as there are in insulin, there are actually billions of possible ways in which those amino acids can be put into sequences. And it's those billions of possible sequences of the 20 different amino acids that gives you the enormous range of proteins that you find in living organisms. Enormous range of proteins, hence enormous range of possible roles and functions that they might have within living organisms. Okay, so if the primary structure is the sequence of amino acids, what do we mean by further levels of structure? For example, secondary structure. Well, having got a sequence of amino acids, and you can see here in this diagram we've got that chain of carbons and nitrogens, not showing the side groups very clearly, but they're hanging off the carbon atoms here. That chain of carbons and hydrogens is then held together into a larger structure known as the secondary structure. And here, for example, you can see a kind of zigzag form, often called a beta-pleated sheet. That structural feature is being held together by hydrogen bonds between the hydrogens and oxygens of the peptide group. Here's another example of a secondary structural feature. So here's the amino acid sequence, and here are the bonds between the peptide groups holding the amino acid chain into a kind of spiral structure known as an alpha helix. So the secondary structure is the folding and coiling of a polypeptide chain to form these structures, held together then by hydrogen bonds between atoms in the peptide group, and it gives rise to these particular structural features known as a helix or a sheet, technically alpha helix or beta sheet. So what then is the tertiary structure? Well, this is a higher level structure. It's the overall large-scale three-dimensional structure of the protein. So if the secondary structure was relatively small structural features within the protein, the overall shape is the tertiary structure. And as you can see here, it's much less regular than those individual secondary structures we saw earlier on. So what is it that holds the protein chain into those structures? Well, as you can see here, it's intermolecular bonds, but this time it's intermolecular bonds between the atoms in the side groups of the amino acids. So here are some of those side groups, and here are the kind of interactions that might hold the chain in a particular orientation. Hydrogen bonds, ionic bonds between charged O minuses and N pluses here, and even covalent bonds in the case of this bond here between two sulfur atoms, which occur between side groups on amino acids called cysteine amino acids. It's even possible, actually, for intermolecular forces as weak as instantaneous dipole-induced dipole forces to help hold the chain structure together. And here this is described as a hydrophobic interaction, but we can regard it as a special case of instantaneous dipole-induced dipole. But the main bonds that you need to be aware of are the hydrogen bonds, covalent disulfide bridges, and ionic bonds. So those are the bonds then that hold together the tertiary structure. So finally, a revision then of the various levels of structure. So the amino acid sequence, showing you in the top of the diagram here. And those amino acid chains can be coiled into helixes or folded into sheets. And that gives rise to this section of the diagram here. And then those helixes or sheets can be held together into a larger structure by the bonds between the side groups in the amino acid side chains. That's the tertiary structure, and it's just possible that several tertiary structures can actually join together to make a protein made up of several subunits. And actually that's a level of structure known as a quaternary structure, but as far as you're concerned for your A-level chemistry, you're not required to know about that. Just one final point about tertiary structure, which will be important later on when we come to look at the factors that affect the activity of enzymes. These weak intermolecular type bonds, particularly the hydrogen bonds and the instantaneous dipole-induced dipole forces, are relatively easy to break if, for example, the protein is exposed to a high temperature. And that can cause the breakdown of this overall three-dimensional structure in a process known as denaturing. We'll learn much more about that later on. Similarly, the ionic bond here can be disrupted if the charges on those groups are altered, for example, by altering the pH of the solution in which the protein is found. You could imagine that at, for example, at low pH, the O- ions here will pick up H+ groups to become uncharged, and that will affect the ionic bond that's holding the structure together. So another factor that can cause the breakdown of the structure of proteins is large-scale changes in pH. That's the end of this video on protein structure. There will follow a further video on the features of enzymes and the factors that affect enzyme activity.